So welcome everyone to the 2023 Ben Collen Memorial Lecture. It's really wonderful to see so many people here, so staff from across the department, from across the faculty. I can see students and also people from beyond UCL. So welcome everyone, it's wonderful to have you here. Before I hand over to John, who's going to introduce today's speaker, I just wanted to say a very few words about the event. So many of you will be familiar with the Ben Collen Lecture. We've been running this now, this is our 5th year. So each year we invite someone, a mid-career scientist who we feel is doing groundbreaking and internationally recognised research in the field of biodiversity and conservation. It's hosted by the Centre for Biodiversity and Environment Research, where we had our former colleague Ben Collen. So this is an event to remember Ben, but also to celebrate all the exciting research that's happening in biodiversity and conservation. We're delighted that this year Marjo Saastamoinen is here from the University of Helsinki to deliver this year's lecture. John will say a few words about Marjo. Before that, just a few housekeeping things. So the lecture around 40-45 minutes. We'll then have plenty of time for questions afterwards. And then we're going to the Haldane room, which is in the main UCL building. Essentially keep going through the cloisters until you almost get to Jeremy Bentham, and then it's on your left. So we'll be going there for drinks and canapes afterwards, so please everyone join us there. There'll be more opportunity to chat to Marjo and to continue discussions. So I'll hand over to you, John.
Thanks very much, Alex. So, yeah, it's a great privilege as Director of CBER to present Marjo, but also to talk about this event which we hold every year to remember our colleague and friend Ben Collen, who I also want to really welcome the members of his family who are here today, particularly Alana, Ben's wife. And this is really a chance for Siberians. We call the members of CBER Siberians. It's basically their chance to nominate. We have a nomination process where people nominate someone they think is doing really groundbreaking work in biodiversity research, someone they'd like to have a chance to spend a couple of days talking to, as I know that Marjo has been talking to many of us in CBER and across GE today. Someone who really is representing some of the key issues in understanding the responses of biodiversity to anthropogenic change, in the hope of perhaps securing a livable future for our children and our grandchildren, and for people across the world. And of course today, the IPCC report came out, which highlights once again the urgency of now and the urgency of science and rationalism in tackling those issues. So thanks very much for everyone for coming today. As Alex says, there'll be drinks afterwards when you can continue to speak to Marjo, and there'll be some time for discussion. But I'm very pleased that Marjo was nominated and selected by Siberians, because I've known Marjo for maybe 10 years or so, something like that. But Marjo actually did her degree in Sussex, and she was privileged enough to be, I think your personal tutor's office was next to John Maynard Smith's when he was still there. So she already has a deep connection with UCL's, one of UCL's most august alumni, John Maynard Smith, and I think you actually had some conversations with him, so that was great. She got her great love of life history, evolution, and trade-offs from those discussions, I'm sure, with him. From there, she went back to her native Finland, to Helsinki, to do her PhD with Ilkka Hanski, another great pioneer in eco-evolutionary research and spatial ecology. Then you did a postdoc with Baz Swan, who also was at UCL, in Linda Partridge's group, back when I started at UCL, and Paul Brakefield in Leiden on bicyclist butterflies and life history evolution. And then back again to Finland for a postdoc, again with Ilkka Hanski, doing really groundbreaking experimental and field-based studies on trade-offs in life history evolution of butterflies in response to abiotic and biotic challenges. And then that was completed with an ERC starting research grant, which now has led on to her being a group leader, again in life history evolution at the University of Finland at the Centre for Ecological Change. And I'm very happy to introduce Marjo today, who's going to be talking about the use of long-term monitoring data and natural populations to understand anthropogenic change. So thanks very much, Marjo. Welcome.
Okay, well, thank you, John, and thank you, Alex, for inviting me here, and for you who, voted for me to give this talk. I'm very honored to give this talk with such a wonderful event. And of course, we all agree that it's very important to highlight also the importance of biodiversity research during these times. I had fantastic discussions today, and I'm going to talk some of the things that I already shared with some of you, but hope you like it. So, like I said, it's becoming very clear that we are faced with global biodiversity crisis and that there are multiple different drivers that are causing species to not do so well, populations to decline, or at least communities to change. So some species, of course, are also thriving due to the changes in their environment. So we often talk about climate change, but of course there's other drivers such as land use, invasive species, and so forth. But all this realization has, of course, catalyzed a demand to preserve biodiversity and develop sort of policies and management plans how to conserve species. But to do so most efficiently, we actually need to know how biodiversity has changed and what are the main sort of causes. So without really reliable quantitative information about patterns and trends, it's actually impossible to conserve or manage biodiversity. And I think that for this, we actually do need long-term monitoring of population, species, or biodiversity in general, and that this should be a resource that many countries have invested in, but there should be even more investments in it. And of course, UK is a prime example of having long-term monitoring schemes, which are still running, and that these are actually the data that we need to show how biodiversity has changed. So here's just examples of different monitoring programs. These can take place at community level, like some of the marine assessments, but also very important data come from just individual, individual-based monitoring schemes as well. So from these data, we are already starting to also see that species and communities are not actually responding equally to the anthropogenic changes. And that is also what kind of is a key interest of my research. So trying to really understand that what factors and potential mechanisms actually determine whether species can mitigate these kind of anthropogenic pressures and are they kind of responding positively or negatively, and what is the strength of the response as well. And this is, of course, a difficult task, but I think it's extremely important way forward. And in this talk, I've split the talk into two different parts. So I will first talk about sort of maybe still my favorite survey. So the Clanville Fritillary Metapopulation Survey in the Åland Islands, where we are assessing the one species within kind of the metapopulation framework, so one big population. I'll then, second part of my talk is more on using kind of these voluntary-based monitoring programs. I'll talk mainly about butterfly work, but also work that has been, is based on monitoring of other groups of organisms. So first about the Clanville Fritillary butterfly and the metapopulation in the Åland Islands. So in Finland, the butterfly only persists anymore in this archipelago, the Åland Islands, which is a group of islands between Finland and Sweden. The butterfly in Finland has a univoltine life cycle, so there's just one generation in a year. And the butterfly is specialized to use these two host plants, Plantago lanceolata and Veronica spicata. These host plants occur in sort of dry outcrop meadows or pastures within the archipelago, and these kind of pastures and meadows are sort of naturally fragmented within the landscape. The metapopulation, so the butterfly has been surveyed since 1993, so now this year is the sort of 30th year celebration for our butterfly monitoring scheme. And it's done via a group of students. So every fall, around 30 to 40 students from the University of Helsinki are taken or hired as field assistants. And we take them to the Åland Islands, where they go through the approximately 4,000 potential habitat patches of the butterfly. And these are defined by the presence of one or both of these host plant species. In this graph, all the dots, the circles, are like the potential habitat patches. The ones that are white are not occupied by the butterfly in this given year. The blue ones are occupied this year, but they were also occupied in the previous year. And then I don't know if you can see there are red dots as well. Those were populations that were occupied this year, but were not occupied in the previous year. So this means that the butterfly exists in this fragmented landscape as a classic metapopulation. So there's a lot of sort of dynamics on which of these local patches in a given year the species is persisting. It exists in approximately 20% of the potential habitat patches, and there's a high turnover between different years. And none of the patches have actually been occupied throughout the whole 30 years. This system was, of course, made famous and well-known, initiated by the late Professor Ilkka Hanski, who wanted to find an empirical study system to actually sort of assess how species can manage to live in fragmented landscapes. And thanks to Ilkka's theoretical work, but then also empirical work on the Clanville fritillary, the system has become sort of a model system for spatial ecology. So we know that this classic metapopulation, which fluctuates, sort of the extinction and recolonization dynamics define the metapopulation persistence. There's a lot of sort of ecological factors that impact these dynamics. So for example, the size of the habitat, the density of the butterflies in the previous year determine probability of extinctions or recolonizations. Habitat connectivity, so how close are nearby persisting habitats, is a key factor. And then also there's a lot of stochasticity. And also this was one of the first natural systems in which it was demonstrated that inbreeding can actually cause extinctions in natural populations. So this was work by Ilik Sakkeri and Ilkka Hanski in the late 1990s. And of course, many of these things have been sort of applicable for conservation biology. So what we have brought with my group to the system, rather than just assessing population dynamics, we've done a lot of experimental work, because it's possible to select a set of populations that you want to contrast to do experiments with. And then you can just go to the wild, collect larvae from the field, and then do experimental work with them. I'm not today, however, going to talk about the experimental work so much, so I'll more focus on kind of some of the information we've obtained from monitoring the natural population itself. So first, talking about how climate warming, or especially extreme events, have influenced the system. So I think the graphs are a bit light, but this first graph, or the two graphs, are demonstrating how in this kind of northern range of the species distribution, the butterfly has actually locally adapted to oviposit in microhabitat sites that, first of all, obviously have higher densities of host plants, but actually those plants tend to be exposed to drought events. And this is not maybe very initiative, like why would the butterfly choose to lay eggs on sites that are exposed to drought? But we've experimentally shown that actually feeding on plants that are somewhat stressed by drought somehow increases also the performance of the larvae. At the same time, these microhabitats are actually warmer and beneficial for the larval development. And so that was the higher top graph shows this preference of the females to lay eggs on drought-exposed microhabitats and plants. In the lower graph, we show that this preference from the female is actually a good preference. So in normal years, choosing these microhabitat sites actually increases the nest survival over winter. So we can also monitor the winter survival, and we've shown that these sites actually are more beneficial for the nests. So this is of course all good. However, now due to climate change, we are more often sort of faced with conditions where the plants don't just get a little bit exposed to drought, but they actually die or become extremely dry, so that there's nothing really for the larvae to feed on. So the picture kind of demonstrates a plantago, which is found on some of these sites that the females prefer, and as you can see that there's not really anything for the larvae to feed on. 2018 was an interesting year in Finland, and in Northern Europe. So this was an extreme dry and hot year, and in this graph, we show with the NDVI, so satellite images measuring productivity, greenness of the habitat patches, that was also reflected on the habitats of the Clanville fritillary butterfly. So you can see the NDVI across the different years, and then the 2018 was this extreme, dramatic decline where there was nothing green in these habitat patches. And this was also then reflected directly to the occupancy of these populations in the metapopulation of the Clanville fritillary, as well as the number of larval nests we found on the occupied patches. So you can see the long-term monitoring data. There's a lot of fluctuations between the years, but the 2018 was a dramatic decline crash year for the butterfly. So this already, of course, highlights that species that are maybe pre-adapted to live in kind of seasons which are short, you know, they kind of prefer these hot microclimates so that their development would be fast enough, then may be exposed to these type of risks if these extreme events take place. Another drastic change that has taken place in the metapopulation is increased synchrony in the dynamics of the local populations. So based on theory, we know that a healthy metapopulation has an asynchronous dynamics. So that means the graph on the top, where some populations are increasing in densities and then others are decreasing. And why this is a healthy metapopulation is that this then allows recolonizations from the sort of okay patches to the areas which are doing bad in this year. And this is how the metapopulation used to be. But then over the last 10 years or so, the dynamics of the system has become more synchronous, which is demonstrated at the graph below. And we've also shown with long-term monitoring data that there is this sort of a spatial synchrony has increased in the population dynamics, and this is driven by climatic conditions also becoming more synchronous within the system. What is actually driving such a synchrony in the climatic conditions in this sort of smaller spatial scale is we don't know that, but it could be that it's something to do with sea conditions in this archipelago area. Okay, so there has been this dramatic crash. What happened after that? Well, the metapopulation actually recovered, or at least that's what it seems like happened. So you can see the decline in 2018, and then you can see the following years. So how is this possible? Well, we don't know entirely yet, and we also don't know what are the long-term effects of this decline. But I will now present some factors that I think make this system actually relatively resilient to some kind of an anthropogenic pressures. I would also like to highlight, which you maybe already see though, that even though the populations have recovered, they are kind of at the lowest level of the previous times. So whether this is actually a full recovery or not, or whether we are also now faced with some sort of different types of dynamics than before, I think we need still a few more years of data to be able to demonstrate that fully. Okay, so first a little bit about dispersal and how that is, we know it is a key life history trait in this system. There needs to be sufficient amount of dispersal to maintain the metapopulation because every patch has a high chance of going extinct. So there needs to be enough dispersal among the patches to maintain a sort of healthy metapopulation as a whole. So based on work that was led by a former postdoc, Michel Dileo, we wanted to use genetic data to try to reconstruct dispersal in this system. So previous studies had used mark release recapture studies, but this is of course often confounded, like you can do it only in a relatively small spatial scale. So Michel used larval samples that we keep collecting during the survey from all nests that we find. She used 5 years of data, and she used this SNP panel that we had for neutral or putatively neutral genetic markers. And she used genetic assignment tests to try to reconstruct dispersal between nests found this year and then nests we found previous year. So trying to assign a family into an individual or family in the previous year's data. And then she was asking how much dispersal is there in this system and what factors are actually contributing to dispersal in the system. She wasn't able to assign a huge fraction of the larvae into a parent in the previous year, just because there's a lot of families or sort of related individuals in the population. So then this becomes very difficult with such a small number of markers. But she was able to assign 30%, and this was almost 800 dispersal events. And we also wanted to be very certain that we were able to assign the parents correctly. And what Michelle showed is that the average dispersal distance of the butterfly is 1.6 kilometers. This is actually exactly what we've shown from the mark release recapture studies before. But what we didn't know before is that there's a lot of individuals who also do very long distance dispersal events. So the longest ones we found from just these 5 years was over 10 kilometers. And she also showed that many females are actually visiting several patches within the networks. So there's actually very sufficient amount of dispersal taking place with networks close to each other. She then also was looking at how different factors such as weather, landscape structure in the matrix, so between the patches, is influencing dispersal, or then the patch characteristics themselves, and then also whether individuals who have different dispersal abilities can sort of transfer or move in a different way. I'm not going to really go into details of this, but just highlighting that for different parts of the dispersal process, so departure, transfer, and settlement, all these variables actually, different variables kind of matter, but highlighting that the quality of the habitat patch, as well as the quality of the landscape in between the patches, is important. So if we are to increase dispersal of individuals among the patches, we need to also take into consideration these type of land use factors. In another study, we've also shown how microclimatic variability can actually be extremely important in buffering against negative things such as mismatches. So a former PhD student, she's showed how early springs in Finland actually initiate the larval development very fast and early. So immediately the snow melts and it starts to be sunny, the larvae start growing and are looking for food. The host plants, for some reason, are not keeping up with this in early springs, and this is actually very bad for the larvae, and we find a lot of starved larvae actually in these early years. So there's this kind of phenological mismatch. However, habitat patches that have a lot of different microhabitats seem to buffer against the negative effects at the population growth level. So we see that it's very important to also maintain microclimatic variability in the local habitat patches. So Michelle also used the genetic data that we had on these SNP panels for over 10,000 individuals collected across the Åland to try to look at whether neutral genetic diversity, so heterozygosity, actually influences nest survival or population performance over the 6-year period that she had here data for. So whether we can find benefits of genetic diversity in this system. And long story short, we couldn't really pinpoint anything in this system, and I'm happy to discuss potential reasons why. So overwinter mortality was lower in nests with increased heterozygosity, but in 1 year only. So in majority of the years, we either found no effect or then even indication for the opposite effect in 2009, even though this was not significant. But just on 2011 did we find a clear indicator that heterozygosity actually matters. At the population level, so trying to see if heterozygosity at the population level influences population performance or extinction probability, we didn't actually find any signal. But then we later also carried out a power analysis, and that showed that we just don't actually have power, even with these 10,000 individuals, to assess that. We did find some sort of indication that the heterozygousity could be more important in predicting extinction probability in small isolated patches. So this is what Illich showed in the 1998 in his paper, but this effect was not significant, but the direction was the same. So that's about genetic diversity. I also will not go in too much detail, but highlighting the fact that the amount of dispersal there is in the system and the stochasticity that we see actually also among the populations probably maintains relatively high genetic diversity in this system. So when we've looked at population or family level genetic diversity over time, there's actually not much sort of fluctuation in it. And also whenever we do experimental work, this is a gene expression study, we find huge amount of variation actually among the different families in the responses at the phenotypic as well as sort of gene expression responses. So the dynamics of the system probably maintain genetic diversity, which maybe is good for future persistence as well. And then highlighting that already during my PhD, we showed that there's also sort of genetic variation in dispersal ability in the system. So we've identified a candidate gene, PGI, which influences flight metabolic rate. And we've shown that sort of these individuals are more frequent in more isolated, fragmented, newly colonized habitat patches. And with Michelle's work, we also showed that these better flyers, who we know from experimental work are better flyers, especially in cooler temperatures, they are also better going through these kind of non-permeable habitats within the landscape. So this also maintains variation in very important life history traits. So take-home message from the first part. The Glanville fritillary metapopulation has or is sort of showing clear ecological changes due to climate change, both extreme events, but also sort of a continuous increases in temperature and especially precipitation. There are probably these pre-adaptations that make the species to some extent vulnerable to especially these extreme events. However, at the same time, the metapopulation is quite resilient, hopefully, as there seems to be quite a good amount of genetic variation in the system, and then also the dispersal ability of the species is kind of maintaining ability of recovering and using a sufficient amount of the patches. However, I would like to note that there is this negative population trend in the system, and what I showed also from before, that whether the dynamics of the system has shifted after the 2018 event still remains to be seen. That if the environmental conditions improve, do we actually see these big increases in population size, or are we kind of stuck now at the relatively low levels of occupancy and population density? So then to the second part of my talk, which is going to be highlighting what we have observed from using these long-term monitoring data sets in Finland. So in the first few examples, I'll talk about the butterfly and moth monitoring scheme data and questions we've addressed using that. So these are relatively long monitoring schemes that people have done in Finland. Obviously nothing in comparison to the British data, but still the butterfly monitoring scheme has been running since 1999 and then the moth since 1993. Even though there's a good data sets, you can still see that of course there's a lot of problems with this type of data. It's spatially, it's not representative of the whole country, so majority of the monitoring is still taking place in the southern Finland. But there's also relatively good amount in the sort of central parts. So in a project that was led by postdoc Maria Helfors, we utilized these Lepidopteran data over 2 decades, and we were interested in assessing potential shifts in phenology as well as shifts in northern range boundaries of these species, and then also linking these shifts to population trends. So in the left side, you see kind of the data points of the butterfly and the moth traps, and then obviously we were looking at the potential range shift from time point 1, so between the 2 decades. And we were specifically interested to see that do species use one, both or neither of these potential strategies to deal with sort of warming climatic conditions. So do species either start occurring earlier in the year, so shift their phenology, or do they move northwards to try to track their climatic niche? Or are species kind of able to do both? Or then neither? And how these potential strategies are actually reflected on the population trends that we are seeing over this same time period. So we found that around 47, so almost half of the species were kind of following our hypothesis 1, which is that they are either shifting their phenology or shifting their northern range boundaries. Actually, very small percentage, so only 13% of the species were able to do both of these strategies. And somewhat surprisingly, almost 41% of the species actually did neither. So they were not able to shift phenology nor their ranges. We included phylogeny also in the analysis, but we didn't really find any clear signal there. And then also we included some traits, but those also had very poor explanatory power. I think we only found the overwintering stage of the butterflies to explain a little bit on potential change in phenology. But how was this reflected on the population trends? Well, we found the strongest positive effect on those 13% of the species that actually had been able to change phenology as well as their ranges, whereas then the clear sort of negative population trends were found on the species that were not doing either. So kind of highlighting that it would be important for the species to be able to trace their these climatic niches in some way, at least, preferably in both ways. We then added to this data some bird monitoring data as well, because Aleksi Leikoinen, that we just talked about, had also found some similar type of patterns. And the bird data is actually a bit better in quality, I think, than the butterfly and the moth data set. So it's been run since 1936 in Finland, and you can see that the spatial resolution is actually much better. So there they've literally made sure that they are covering the entire country, even though the northern parts are still often a bit less frequently assessed. So in this study, we were particularly interested to assess whether sort of past selection to climatic conditions would shape how species are now responding in terms of range shifts. So do attributes describing species' climatic niche actually explain how they are now responding? And for this, we use something that is called species temperature index, just because sort of it's difficult to know the thermal performance curves for all of the species. So we use the species temperature index, which is an average, or we use the average and the breadth of this measure, which is calculated from the temperature that the species is experiencing within its European range. So this was kind of used as a proxy of the species' thermal niche. We also focused only on sort of the southern species, so those species that would have their leading edge in Finland, because we were only using monitoring of Finnish data. So we removed all the species that were kind of going to Norway. And then we calculated the realized range shifts over the last 20 years. So we found that the mean shift was actually highest for butterflies, so over 40 kilometers over this time period, followed by birds and then by moths. And maybe somewhat surprisingly, we found that actually the relative niche breadth did influence the likelihood of range shift, but in a negative way. So those species that had more narrow thermal tolerance were actually following the climatic condition in the range better. And this was evident for birds as well as the moths. So whether this is due to the fact that those species kind of are more, they need to follow their climatic condition more because they have much narrower tolerance, or whether these species, for example, also have higher dispersal ability or ability to seek for thermal conditions that are good for them, we don't know, but would be potentially interesting to assess. In the moths, we also found that species with warmer thermal niche tended to shift northwards more. Again, we included traits, but some had mild effects, but nothing extremely traumatic, except for maybe the sort of migratory or resident species. Butterflies showed, kind of significantly, they showed nothing, but if you look, they actually showed a bit of an opposite trend. So in butterflies, we found that niche breadth, but actually only the moisture niche breadth. So we didn't only measure the temperature, but also precipitation and moisture. There we found that butterflies that had a broader niche breadth for moisture actually were maybe moving northwards more. And the temperature effect, or generally kind of the range shifts in the butterflies, we are thinking that it could be that they are more constrained by land use and available habitats in Finland, and this would be a very important thing to assess in future studies. Okay, so I keep adding species to my figures. So in then the next analysis, we decided to go and add even more. So we took into consideration also winter track monitoring, which is done by hunters in Finland. So this gives us data on mammals. And then also there are small rodent monitoring schemes. And then also plant, so vegetation assessment. Oh, and also phytoplankton, so some aquatic things as well. So this is something that within the Research Center for Ecological Change we have in Finland has been kind of our goal, that we wanted to bring together all these different types of monitoring scheme data and then analyze obviously different species groups separately, but still have kind of same framework for the analysis to try to understand are species showing similar or different patterns over time. So these are the different data sets, and this study was led by fantastic Laura Antao and Ben Weigel. So we were interested in trying to understand how the biodiversity or these communities had changed over time, now over 4 decades if possible. And then we also looked at different drivers. So we had temperature, precipitation, snow cover, and then the northern Atlantic oscillation as potential drivers of these shifts. Because of the time, I'm only focusing on the data on temperatures. So we were asking how communities had changed over time, and then more specifically, if they have changed, how have species actually potentially shifted in their kind of relative niche space. So we were interested to compare between these time points whether species are moving towards kind of optimal niche space due to climate change. Are they at their optimal already and just stuck there or happily there? Or are they actually shifting over to kind of decreasing, that they are now kind of outside of their preferred niche space? So first, like I said, I'm now going to just show data or results on temperature, but this was actually the driver that had the highest explanatory power, followed by snow duration, which makes sense because, of course, snow it's very important in Finland, and there's a lot of changes happening in the amount and length of snow. We found actually quite a bit of turnover in these communities, but highlighting that still within the climatic zones, the species that changed were still sort of shared. So there weren't really new species coming into these communities within each climatic zone, nor did we lose species, but still kind of at the different spatial locations, the species were shifting quite a bit. And we also saw a lot of shifts in this kind of a niche space of the species. So this is a bit of a complicated figure, but this now shows all the different data sets. So we analyzed the different groups of organisms separately. And then the numbers, so this is for the northern boreal zone where we saw biggest changes in the community, so in the north. And then the 1, 2, 3, that means the decades, so the first, second, third, or the fourth decade. And for the small rodents and butterflies, we didn't have now, I'll show another graph soon, but we didn't have data for this. And then the colors represent part of this niche space that the species, proportion of species were in these different decades. So, for example, if we look at the birds, we can see that increasing amount of species were actually, becoming kind of increasing their optimal space position in the north. So the environmental conditions, the temperature is being better for the species occupancy. However, there's also increasing amount of species in the birds which are shifting over, so actually are now exposed to thermal conditions which is not anymore optimal to them. But generally these patterns were also quite variable among the different species groups. So this is now for the north, and then I can just show you how it was, for example, for the south. And here the situation is a bit different. For something like butterflies, it seems that even in the south, the conditions are actually becoming suboptimal. So they are already shifting over the optimal space, and similarly to some of the mammals at least. We haven't yet looked at the responses at the species level, and this would of course be interesting to see if there are sort of traits or factors that explain why or what groups of actual species within these groups of communities are responding the way they are, but that will hopefully be the next stage in this analysis. I don't know how I'm with time, but this is kind of okay. Sorry, I was so nervous of whether I'd make it to 40 minutes, so maybe I've talked a bit too fast. Okay, so the final bit is very recent and kind of in review project, which was led by another postdoc, Andrea Santakelli. And in this work, we again used multiple of these monitoring data sets, and we were again interested in how communities have changed, but now particularly whether the patterns of change differ if the monitoring, the species occur within protected areas or outside protected areas. So trying to understand how effective actually protected areas are on changes in occupancy patterns. So in this graph, you see protected areas, like transects or data sets from protected areas, and then those from unprotected areas. And the green, which you see much in the north, highlights actually where the protected areas in Finland are. There's a lot of protected tiny areas underneath the black dots, but you just don't see it at this resolution. So this also kind of highlights, I guess, generally the problem with protected areas in Finland, that they are all in the north where, rather than in the south where probably species would need it more. But in any way, we did this analysis, and of course we understand that there's a unbalance in the data. So what we did is that we used this counterfactual matching approach to account for the land use in a, kind of a buffer area of these transects. And then we found like a matching site for every sort of protected area with an unprotected area. And then we did this matching within each of the climatic zones. So the pairs weren't like from the north and from the south, but always from the same geographical area. And then we looked at occupancy patterns of approximately 600 birds, mammals, plants, and phytoplankton. And as you can see, we had to drop butterflies and moths, and this is because there's no monitoring actually taking place in the protected areas in these species, so we couldn't include them. Also, bear in mind that we are now using only kind of the common species. So because of these long-term monitoring data sets, the rare species have to be excluded from the data because we don't get really reliable information on their occurrence patterns. So these are kind of species that are still abundant in Finland. Okay, so this is what we found. We actually found that for majority of the species, the protected area, it doesn't matter like at all. It's only for quite small fraction of the species, 20% for birds, 13% for mammals, and then less than 5% for plants and phytoplankton. Are the species doing better within the protected areas and not? And I really don't want to say here that we shouldn't protect areas, and that's what I'm going to come back to in the final parts, because of course, conserving areas is crucial for many, many species, but we are not directly targeting, obviously, those species with this analysis. But let's see, so what does this mean? So we also wanted to try to understand what is that is either making the effect positive or negative for the species, to try to understand a little bit more what is going on. And these are kind of the different hypotheses that we had. So protected area can have a positive effect compared to the unprotected area if things are stable in the protected area but declining outside, so in the unprotected area. It could be also that the increase is just more stronger within protected area than from outside, or then there's totally like contrasting patterns. And then the negative effects, of course, would be just the opposite, that the species are doing somehow worse within the protected area than outside protected area. And this is what the graph showed. So now if you go at the species level from the 0 point to this side, that's the positive effect. And then to the other side, it's the negative effect. And then the colors are now representing which type of these responses were kind of driving this positive or negative effect. So most of the positive effects, as you can see, are yellow in majority of the groups. And this means that we found slower range of declines within the protected areas among species that were actually overall declining in Finland over this time period. There's also some red, especially in the birds and the phytoplankton, so showing that the increase is stronger within protected areas than outside. The negative effect, on the other hand, seems to be mostly red, maybe not for the plants, but for the other groups, which means that these are actually generally increasing species in Finland, but the increase is just less prominent within the protected area. And I think here we now need to move step further in our next analysis to try to understand what these species, or assess the species actually more kind of individually, because of course these are most likely the generalist species that are just doing extremely well in Finland and are just sort of spreading everywhere. And the negative effect that we're now saying could actually be a positive effect, meaning that the protected areas are buffering these species to kind of take over the community or enter the community. So, yeah. So, I know I really want nobody to take this message wrongly. So, what I think this result means is that protection for sure is good. There's a lot of evidence showing that we need to conserve, and conserving 30% is definitely what we should do, even more. But highlighting that protection and conserving habitats alone is not actually enough, that there's a species even outside protected areas that are not necessarily doing well, or communities are shifting also, in these other habitats that we are not, at least in Finland, protecting. And hence, we have to make sure that the quality of those habitats, their connectivity, is enough to actually halt the biodiversity crisis. So if you say anything about this, please remember this statement. Okay, so then take-home message from the part 2. So it is evident that some species are sort of winners and are increasing or doing better in space or time, whereas others are losers. Unfortunately, I don't think we are even close of trying to pinpoint who are those species, that are winners and who are the losers and how do we define them. So much more research on that front still needs to be done. However, I do think that we probably have to move beyond these sort of traditional traits that we have in trait bases like body size, generation times, whatever, that we may have to think a bit closely of life histories. Well, generation time definitely is an important life history, like dispersal, thermal tolerances, these type of things. So think a bit beyond kind of the most obvious traits. And then also, simply by conserving areas, we probably will not halt biodiversity loss. So we need to make sure also that areas outside protected areas are, to some extent at least, good and connected to each other. And with that, I would like to thank you for listening. Again, thank you for giving me this opportunity. I want to thank our funders, the people who have provided these long-term data sets. They come from different research institutes, universities, and so forth, and then a bunch of collaborators that have been involved in this work. Thank you.
Thanks very much, Maria. That was wonderful. We have, I think, time for questions. So does anyone have any questions for Maria? Yes.
That's great, thank you. I work in policy on protected areas and other things, so that last was really interesting. So I don't think I missed it, but did you look at the effect of size of protected areas? So you looked at pairs of them, but presumably most of them look very small, so they're going to have major edge effects and things. So in the same way, the sort of metapopulation part, have you put the two together to look at what kinds of protected areas were affected?
No, we didn't. This was a really crude I was supposed to repeat the questions. So the question was whether we accounted for the size of the protected area or type of a protected area in the analysis. No, we didn't. And we actually, some of the transects were not entirely within a protected area. So now I don't remember how much. I think they had to be like 70% or something within a protected area that we considered them to still be within a protected area, assuming that there would be some sort of a spillover effect maybe from the conservation area. But no, we didn't in this one. So this was a pretty crude, it was still kind of habitat matching. So, they needed to have still for a similar type of a habitat surrounding them. But for sure, obviously, the size of the protected area could matter.
Would the metapopulation analysis give you any hint that
Yeah, definitely. Yes, exactly. Yeah, so you would think that the larger areas, and of course those that would be maybe connected to other protected areas. But I think by starting to do that, we would probably lose power, because already with this matching, we get, you just start kind of having less and less things that you can match. Yeah, exactly. Yeah, that they are all in the north where, yeah. So repeating the questions, if I remember, the first one was about the resilience of the metapopulation. So how do I think how long would the resilience sort of last and when would it break? I don't know. I think it would definitely be interesting to indeed see whether it's now just holding on to something and then will crash. There is one study from Ilkka, using genetic data from museum samples of the mainland Finland, where the butterfly used to persist, but then it went extinct, where they showed that the genetic architecture of the species prior they went to extinct was looking similar to our more fragmented, isolated areas, where we had this higher level of these good dispersers. So kind of those individuals seem to be selected for, because the habitat probably was getting more and more fragmented in the mainland, but then that wasn't enough, and then the population went extinct. So I think this is one of the things that we could try to understand, that we now know the dispersal distance. So of course you could model like how less connected can the patches become before there's actually not sufficient amount of dispersal between them to maintain all these fluctuations. So I think this would be how I would maybe start doing it, but we haven't done it. And then the second question was, yes. So the butterfly in Finland is univoltine. In Spain and France, there are also bivoltine populations, but also at least in Spain, there are also univoltine populations, and this is weird that they seem to not be, at least in many places, sort of mingling, that they are separate. But anyway, so that means that maybe there is at least, some potential that it could become bivoltine, but that's pretty far because it's still those French populations. We do see sometimes the larvae are trying to skip the diapause, but then they, for some reason, tend to die. And then, of course, the species doesn't really occur more north, so that will be there's not really habitat patches available because it requires areas which there's enough grazing. I think another potential adaptation could be the site choice, where females are laying eggs. So if they would start preferring not these really hot open areas, but actually more shaded areas where the plantago is still growing. But I don't actually know if this site choice has a genetic basis, or is it more like bed hatching strategy. So we don't know, but those again could be.
Yes, referring to the collapse in fertility in 2018 and recovery in 2019, I wondered what the effect of predator species was on that, and whether predators had also collapsed, and that was part of the explanation as to why there was a recovery in 2019.
So the question was whether the collapse in 2018 was, or the recovery was potentially influenced, that also the predators of the species maybe collapsed, and then them not being there, there would be a recovery of the population. So it's interesting hypothesis. So we don't know actually predators of the species. They feed on these plants that have secondary compounds, these iridoid glycosides. So at least birds don't eat them. So they taste really bad. But there are definitely some insects that can still be feeding on the larvae. We do know parasitoids of the species. So there's 2 specialist larval parasitoids that actually can have quite a big impact on the dynamics of the species. One of them is extremely local, and the person who is studying the parasitoids was really worried that the parasitoid would go extinct because it's very rare. It didn't. So the parasitoid is still there. We don't know. I think we don't have enough data. So we collect larvae every year from the field, and then we rear some of them in the spring. I think we haven't looked at whether the parasitism level of the other species, this hypersoter, which has been 30% before, whether that would actually be lower nowadays. Or would have been in the previous kind of the early years. So that could be an interesting thing to look at. But other than that, we don't know about the predators.
Any other questions? Yes. So the glandular system, you have a really good understanding of what's going on there. But it's obviously restricted in space. And then with the monitoring scheme, you've got this massive northerly gradient, but incomplete data. And so as somebody who works with large-scale long-term data sets, how should we be changing the design of our long-term monitoring schemes in order to better answer the questions that we'd like to ask?
So the question was about how we could improve the long-term monitoring data to actually have more sort of better ways of observing these responses, because there's a lot of biases in them. Well, I hope in a year and a half I can address this better. We just got a grant from the Ministry of Environment in Finland to address this. So we are aiming with the statistician, Jarno Vanhatalo, who is a fantastic statistician, trying to assess that what can we actually say with the current monitoring schemes, and then maybe what we cannot say, and then how could we improve it, to cover with the amount of monitoring we can do, so kind of realistically, but how could they be spatially maybe better located or temporally, that we would get most out of it if we are to understand biodiversity change. Because I think also many of the monitoring schemes, that's not the reason why they are doing it. So, but if we would be, monitoring biodiversity, how it should be done, that's what we aim to assess.
Maybe a final question, if there is one. If not, I think that means there was one. So that's it.
For your butterfly populations on these islands, are you concerned that if there's another drought effect, the population might crash? Saw that almost in 2018. Do you think that you would want to add some watering or some mitigation measures to help try to preserve the species?
So the question was whether I'm worried that there would be another of these drought events, and that the meta population probably wouldn't sort of survive on its own, and whether there should be some interventions of trying to rescue it. I don't know. The species is still existing elsewhere. It's also doing quite well, or improving actually in somewhere in the Central Europe. I don't know if I would do watering. That would just be too much maybe for this whole area. We're barely able to keep the monitoring program going. I think it would be even more difficult to then do that. But maybe trying to think if there's some other things we could do, like maintaining some of maybe key habitats or networks, make sure that they are grazed properly, that there would be habitat heterogeneity or something like that could be done to just suggest some improvements of some of the areas maybe that could then help. And I do think that, I don't know, there's a lot of nests still around and a lot of populations occupied, but the drought event did cause extinctions of some of the populations in these islands. So the map that I showed, it's still kind of these big mainland areas that are sort of interconnected, but then there are smaller islands that have had populations persisting, and actually we've lost those ones. So there's been extinctions of some of these areas. So I think there is a risk of that happening in some of the areas.